Hello, welcome to 13 Universal Learning Beyond Barriers. This is CIE A-Level Biology, and we're going to be looking at all topics series 2. This is Shedro, your tutor. So let's dive into it. So we're going to be looking at all the topics. We have done series 1. Okay, let's look at question 1. It says which molecules contains at least 4 double bonds? Okay. So which of them contains at least 4 double bonds? Unsaturated triglyceride, hemoglobin, collagen. Which one do you think? Pause the video and try to solve it. Okay, but there's one I want to tell you. For unsaturated triglyceride, remember that triglycerides, basically triglycerides, okay, it's made up of one glycerol, okay, and 3 fatty acids. So 1 glycerol and 3 fatty acids. Remember that the 3 fatty acids, each of them have each of them has what we call carboxylic group. Is that okay? So if you look at this now, this is one of the fatty acids. This is a carboxylic group. Okay, yeah. So if you look at this is yeah, carbon to carbon chain, hydrocarbon chain. I'm just using this to represent it. Okay, if you look at this one also, you will see something like this. Okay, now this is the ester bond I'm drawing. Okay, so watch this. This is another fatty acid attached to this triglyceride. You can see that. Now this is hydrogen carbon double bonds with oxygen. So this is hydrogen hydrocarbon chain. All right, so maybe one of them or two of them has saturated hydrocarbon chain, but the question says that we're dealing with 4 double bonds. Now look at this. Watch. So let me draw the third one. You can see the third one here. So you can see. Let's assume that this is another carbon oxygen double bond. Okay. Now this is another hydrocarbon chain. Let's assume we have only one double bond. So I can put one double bond here. Yeah, that's it. Let's assume one. Okay. So stay with me. Now I should put hydrogen here to make this glycerol. So this is ester bond. Ester bond. There are 3 ester bonds here. This is triglyceride, right? We're looking at unsaturated. Let's assume one of them is unsaturated. That's the presence of double bonds. Okay. So if you look at this, we're looking at which molecules contains at least 4 double bonds. At least 4. So this is a double bond between carbon and oxygen. 1. This is another 1, 2. This is another 1, 3. This is carbon, double bond here between this carbon and this oxygen. Do you get? So just the fatty acid alone gives us 3 double bonds. Now because they said unsaturated, all right, you see it unsaturated. So one of them, let's assume one of them is unsaturated, that will make it 4. If 3 of them are unsaturated, that will make it 6. So unsaturated triglyceride should contain at least 4 double bonds. 4 double bonds. Okay. So if you look at collagen, now guys, don't forget that collagen, look at collagen here. You will see that collagen or hemoglobin, they are all made up of amino acids. So let's look at, if you remember, the structure of amino acids. So amino acid is made up of a carboxylic group, which also contains carbon double bond oxygen. And of course, this is an OH here. See? This is OH. You can see OH here. Okay? So this is amine group. So every amino acid has an amine group, a carboxylic group. All right, you can look at this. We have this, okay, this amine group, and of course we have R group. R group is what is different in it. So this is hydrogen. Stay with me, don't go away. Okay, so watch guys, see, we have this. Every amino acid contains a carboxylic group and amine group, right? an aryl group, and of course this hydrogen. Now this carbon here that every other group attaches to. This is alpha carbon. Okay, so if you look at this, you see that at least there's one double bond, at least one double bond in every amino acid. That's carbon, double bonds, oxygen. Hemoglobin is made up of many amino acids because it has 4 polypeptide chains. Each polypeptide is made up of a lot of amino acids. Okay, so that means you're going to have more than 4 double bonds in hemoglobin. The same thing with collagen. Collagen is a protein made up of amino acid. So it has 3 polypeptide chains. Each chain contains more than 4 amino acids. Do you understand? So this will contain at least 4 double bonds. This will contain at least 4, of course. This will contain at least 4. So D is the answer. So all of them contain at least 4 double bonds. Please, would like you to share this video, okay? Make comments and let us know what you think. So what is the answer now? The answer is D. All right, let's go to the next question quickly. The diagram shows the amino acid and glutamic amino acid. All right, glutamic acid. That's the glutamic acid. That's amino acid. That's what we're talking about. Okay, this is glutamic acid. What is the aryl group for glutamic acid? So pause the video. All right, and try it. What is the aryl group here? Remember, please see the secret to this. Every amino acid will always have a carboxylic group attached to the same carbon atom as the amine group. So this is the main carboxylic group. This is the main amine group. Is that okay? So every other thing by the side is a side chain, aryl group. So this is the aryl group. Aryl group is this chain here. Okay, it's not the main carboxylic group. So this carboxylic group here is not the main. So it's just part of aryl group. So if you follow me, this is it. So this is aryl group. Okay. So how do I know? Because you must be able to identify the main carboxylic group, which is always attached to the same carbon atom as the main amine group. So both of them, this is terminal carboxylic group, which is the main carboxylic group. This is terminal amine group. Okay, so they are both attached to the same carbon atom. Once you identify 2 of them, other groups, right, still attached to the same carbon is the aryl group or the side chain. Okay, so the side chain, of course, you know what it is. It is D. All right, let's go to the next question. The diagram shows 3 interactions that hold protein molecule in shape. So look at that. You can see that. So what are the interactions? Okay, what this is trying to say, this is typo. What interactions? What are the interactions X and Y? What are the bonds X and Y? Okay, so what do they represent? Okay, so if you look at it, let's look at X, the interactions. All right, that's talking about the bonds here, the bond here, and the bond here. This is a typo, but what we're looking at here is the bond present. Bond present. Okay, so see interaction 1. Interaction 1 is this bond. So this bond, you see this bond is formed between the hydrogen of an amine group and the oxygen of a carboxylic group. This bond here, all right, is hydrogen bond. Hydrogen bond is always formed between the hydrogen of an amine group and the oxygen of a carboxylic group. Okay, now if you look at this is a disulfide bond because it's formed between 2 atoms, 2 sulfur atoms. All right, now if you look at interaction 3, it is formed between ionized, this is amine group, but it's ionized amine group and ionized carboxylic group. That's ionic bond. So interaction 1 should be hydrogen bond, so we have A and B. All right, interaction 2 should be disulfide bond. Okay, but if you come to interaction, if you follow this A, now disulfide is also covalent bond, so 2 of them are correct. Okay, it's also covalent, strong. Then if you come to interaction 3, hydrophobic interaction, that interaction is not hydrophobic. Hydrophobic interaction is interaction between non-polar aryl groups. Is that okay? And of course, it is ionic bond. So hydrogen bond formed between hydrogen of NH group and oxygen of a CO group. Okay, covalent bond or disulfide formed between sulfur atoms, and then ionic bond formed between ionized amine group and ionized carboxylic group. Is that okay? So the answer is what? Is B, right? All right, let's go to the next question. So some animals produce antimicrobial proteins which protect them from pathogens. That's important. There's defense mechanism, guys. Okay, I want you to pause this video. I won't get to a point of answering the question and try it. Engage with us. These proteins could be used to kill human pathogens. However, when used as a medicine, they are broken down by protein digesting enzymes. Yes, our protein digestive enzymes will break them because they are also proteins, like the pepsin in our stomach can break them down, or trypsin in duodenum, or epsin in small intestine. All those protease enzymes could digest it down. They now said replacing one of the amino acids found in the protein with an amino acid that had been synthesized in the laboratory resulted in a modified protein that was not broken down. So this protein could not be broken down. The enzymes could not break the protein down. Okay, so they said what could explain why this modified protein was not broken down by the protein digestive enzymes. Okay, let's see how it happens. Now, number one says modified protein. Okay, let's list it. Now, I want you to check which one do you think is the answer, right? Which one do you think is the answer? But let's work it out. Remember, guys, if you're conversant with enzymes, okay, remember that enzymes have tertiary structure, okay, which is their active site, all right? Yeah, and also don't forget that substrate also has tertiary structure in their protein, okay? So this is the substrate that is acting as medicine, right? This is enzyme E, all right? So guys, if the shape of this, remember, there's the question here says what could explain why this modified protein was not broken down by the protein digestive enzyme. This is the protein digestive enzyme. This is that protein that's used as medicine before it could break it down because it's complementary. Okay, the substrate could bind to the active site and then due to interaction it's broken down, but something happened. The shape change. Things happen, people change, things change, something happens. Maybe when they change the amino acid, the shape becomes like this. It becomes like this, all right? Okay, instead of this, all right? So this may not be able to bind to this, okay? So they're not asking us why, but this is tertiary structure. Okay, the tertiary structure is what changed. They now said what could explain why this modified protein was not broken down by protein digestive enzymes? Number one says the modified protein has a different tertiary structure to the original protein. Yes, you can see that the shape has changed from here to here. One is correct. Okay, you're no longer fitting. Number two says the modified protein not complementary in shape to the enzyme's active site. Can you see? This is no longer complementary. Do you see? Two is correct. Three says the modified protein is unable to induce fit with the protein digestive enzyme, so it's not able to induce it. That means this is no longer able to cause it to change shape, because if it's able to cause the amino acid, the active site to change shape to break it down, it could work, but it cannot work. All right, so it's not able to induce a change in shape of the active site. So that means that the three options are correct. So the answer is A. All right, let's go to the next one. Now, a student investigated, please forgive this, the student investigated the effect, this is effect, please, of substrate concentrations on the rate, this is rate of an enzyme catalyzed reaction. A graph was plotted to show the relationship between these two variables. The student was asked to take readings from graph that could be used to determine the Michaelis-Menten constant Km for this enzyme. So you can see JKL and M show points read from the graph which the students could use to determine the value of Km. Okay, so this is the graph here. So which two readings must the students use to determine the value of Km? It's must the students use. Okay, must be used by the students. All right, so now which one must be used? So to determine Km value, guys, remember, if you remember the theoretical concept behind this, so Km value, remember that Km, which is a substrate concentration at half Vmax. Okay, let me let me draw it again. This is Km, okay, is equal to, right, that's substrate concentration at half Vmax, at half Vmax. This is Vmax. Vmax is the maximum rate of reaction, okay, so over 2. Okay, that's the formula of Km. So if you want to determine Km, first of all you have to know the Vmax, okay, and then when you get the Vmax and then divide it by 2, this is 2, right, then you come to half Vmax. Like if this is Vmax, half Vmax in this graph is L. So because if this is Vmax, half is L, okay, if J is a Vmax, half is L, so you have to know L must be there. Then you now trace this half Vmax to the graph, because Km is substrate concentration at half Vmax. Km is not half Vmax. It's not actually half Vmax. And you know, substrate concentration is on the x-axis. So what you do is that you trace it, okay, and then extrapolate it down to substrate concentration, and the only graph that shows that is K. That's the only part that shows that. So we need L, we need K, we also need J, but if we check 3 of them, the most important ones there is L and K. Okay, so L is half of the Vmax, K is a Km itself because it is half, all right, it is substrate concentration at half Vmax, all right. So K, L and K, where are they? Where is K and L? What do you think is the answer there? Okay, K and L, okay, because this is half Vmax, this is substrate concentration, so the answer is C. All right, let's go to the next question. So the end product of metabolite pathway can act as a competitive inhibitor. That's end product control mechanism. This is called end protein inhibition and allows a cell to control a metabolic pathway. The diagram shows a metabolic pathway where the end product could act as an inhibitor of enzyme W. Guys, they always asking this question, right? Okay, let's see. Let's say what would be the effect of if enzyme Z, this is enzyme Z, was inhibited by the end product instead of enzyme W. Okay, if it was inhibited by the end product, if this enzyme Z is inhibited, if it's inhibited by the end product, okay, instead of W, instead of going this way, this inhibits this. So if this inhibits this, that means that production of this will stop. So okay, let's go to end product. Which option says that it will decrease? Because once this is inhibited, remember that this enzyme catalyzes formation of end product using intermediate 3. So intermediate 3 is converted to end product by enzyme Z. When enzyme Z is inhibited by end product, that means the product of the amount of end product will be reduced. Is that okay? So A and C, they are most likely. So we're eliminating B and D. All right, let's check. Of course, the next intermediate 1 is converted from here. Remember that substrate is being converted from intermediate to intermediate 1. So W was not inhibited, so this will increase. Okay, intermediate 1 will increase because the enzyme producing it is not inhibited. So why this increases, this decreases. So which option? Okay, the option is A. All right, let's go to the next question. Which row shows the distribution of cholesterol and carbohydrate chains of glycolipids and of course and glycoproteins in a cell surface membrane? Now cholesterol is between the phospholipid, because remember guys, cholesterol has if you check the phospholipid bilayer, just permit me, you know, phospholipid bilayer appears like this, okay, and you know that cholesterol has similar structure as phospholipid. It has hydrophilic head and hydrophobic tail, okay. Yeah, that's bilayer. So let's assume I want to put another one here just to paint the picture for you. Okay, this is a phospholipid molecule. All right, this is also a phospholipid molecule. Here I'm drawing this. Okay, you see, because cholesterol has the same structure, so cholesterol will align. Now let's say this is cholesterol, all right. Okay, okay, with its tail, so it will align Now, if this is cholesterol, guys, it will be within the phospholipid, so that the hydrophilic head of the cholesterol molecule face in the same direction as hydrophilic head of phospholipid. Okay. The same thing with the tail. The tail faces the same direction with the tail of phospholipid. Okay. So that's what is happening there. And of course, it will not be. This one says between phospholipid head only. No, it will be between phospholipid. So the carbohydrate chains are usually pointing outward because they help in acting as, of course, cell markers. So they form hydrogen bonds. So if you check at the carbon, the polysaccharide carbohydrate chains, they always point out. Like for example, this is phospholipid. This is chains of glucose attached to phospholipid molecule. If it is glycoprotein, this is a protein. Protein is present like this, and we have the same chain. Okay, yeah. So these chains point outward. This is inside the cell, and that's outside. All right. So it points outward. All right. To form hydrogen bonds with it. You get? All right. Hydrogen bonds with water, so they point outward. So A and B is eliminated because it must be between phospholipid. Okay. Mainly on the inner side. I think it's mainly on the outer side because it forms hydrogen bonds with water to stabilize it, and that is where most, of course, antigen bind to, because it acts as for cell signaling, or where it binds to other molecules for cell recognition. So cell recognition, yeah, especially for glycolipid. But glycoprotein acts. It helps for cell recognition and cell signaling as well. So they are found on the surface. Okay, so this means this is likely going to be the answer, right? So this is the answer. So let's hear your comments. Let us know what you think. Which rule correctly describes all the possible relative concentrations of substances when the substance is moved by endocytosis or exocytosis? So let's look at it. Okay, now this, don't waste your time here. See, there's just information you need. Exocytosis or endocytosis, they are not affected by concentration gradient. They can move against concentration gradient. They can move down concentration gradient. They can as well move when the concentration gradient both outside and inside are equal. So if you have something like this, we have this as cell. Okay, so endo is moving inside, bulk molecules. Exo is moving outside. So molecules, if you're moving this molecule X from outside, from inside to out, that's endo. Exo, sorry. This is Y is moving in by endo, from outside to inside. X is moving, so it's not affected by concentration gradient. It's not affected, so it can go either ways. So this concentrations equal, it doesn't mean it mustn't be equal. Concentrations equal, greater, yeah, this is the answer. Don't waste your time on this question. All right, that's it. Okay, so let's go to the next question. Which statement about simple diffusion is correct? All right, pause this video and try to solve it. Now, it requires specific molecules in the cell surface membrane. It doesn't require, because simple diffusion involves movement of non-polar molecules. Now, B says it is a passive mode of transporting substance. Yeah, it's passive. That's correct. C says it always requires a membrane for transport of substance. No, it doesn't. It doesn't always require a membrane. All right. It always requires a membrane for transport of molecules. The molecules can actually diffuse. They're passive. All right. This says it only happens in the cells of prokaryotic and eukaryotic. Okay, it happens in all cells. Okay, all right, even okay, it happens. Okay, so that's not we're looking at which statement about simple diffusion is correct. So it is passive mode of transporting substances. That's the correct one. So it is D. Let's look at the last question. Now, plant cells with the same water potential in their cytoplasm were each put into one of the three concentrations of sugars. This is the highest concentration. Okay, so any solution that has this 10%, don't you think it will have the lowest water potential? Remember, the higher the water potential, the lower the solute potential. The higher the solute potential, the lower the water potential. So solute potential and water potential are inversely proportional. So this will have the lowest water potential. All right, this will have higher water potential than this. This will have the highest water potential. They say the cells were left for 15 minutes and then observed using a light microscope. So when you observe it with light microscope, they say which statements are correct. Okay. Now, sorry for the answer appeared before we analyze it, but let's look at it. This one says cell Y had a lower water potential than the sugar solution. Yes, if you put a cell in a solution that has lower water potential than the of course than because I say Y, so cell Y had lower water potential than the solution. So because it has lower, so water will move from solution into it. So this becomes bigger, turgid. Yeah, that's correct. So water moved in from solution into cell Y because cell Y has lower water potential than the solution. So water moves into the cell. Anytime the cell has lower water potential than the solution, water will enter the cell by osmosis. Is that okay? 3 and 2 says cell Z was put into 10% sugar solution. Okay, look at cell Z. Yeah, most likely it has higher water potential. This is plasmolized. This cell is plasmolized, right? So that means it lost water. Okay, so that means it was put in a solution that has lower water potential. So it lost water, moved out of it, and it became plasmolized. So 2 says cell yes. So it was put in the one that has lower water potential, and you see 10% has the lowest water potential because it has the highest soil potential. Now, so 2 is correct. 3 says cell Z had a less negative water potential than sugar solution it was put into it. Yes, less negative means low water potential. So it has low water potential, okay? That's why it lost water. Cell has low water, it has, because they say cell Z had a less negative. Less negative means, less negative means higher water potential, yes. So cell Z has higher water potential than the solution it was put into. Less negative means high water potential. More negative means low water potential. So this has less negative. That means it has higher water potential than the solution it was put, sugar solution it was put into. So it lost water from high to low. So all of them are correct. All right, so Asia, so thank you guys for joining us. Please, we want to see your comments, share the videos. All right, so very soon we'll start with the next series, which is series 3. All right, see you guys this schedule. Bye.